Good morning, everyone, and good evening to those who are watching us from different time zones. This is our very first webinar for 2024. I'm Kathy Skopelitou, the host for today's webinar, and today we have the great pleasure to have an exceptional speaker with us today, Professor Rui Moreira. But before I give the floor to Rui, I would like to share with you a few information about our research infrastructure. EU-OPENSCREEN is a European research infrastructure consortium for chemical biology and early drug discovery. We support all stages of a chemical tool development project, including assay adaptation, high-throughput screening, and chemical optimization of the hit compounds. Furthermore, EU-OPENSCREEN operates an open access database and a unique common compound collection. Our main library, the European Chemical Biology Library, consists of over 100,000 compounds. EU-OPENSCREEN has over 30 affiliated high-throughput screening and chemistry facilities at partner sites in 9 European countries. These partner facilities provide researchers with access to cutting-edge technologies to develop their own tool compounds. So if you are a biologist or chemist and you would like to work with us, you can find more information on our website. If you click on the Participate tab, you can find here these links: access for biologists, assay providers, and access for chemists, compound providers, where you can find more information about how you can work with us. We have also some very informative videos that you can watch in order to see the way you can have access to our research infrastructure. Okay, back to our webinar now. The webinar topic for today is from Professor Rui Moreira about the novel chemical tools to target ferrous iron-dependent biology. A few words about Rui. Rui is a professor of medicinal chemistry at the Faculty of Pharmacy in University of Lisbon since 2006. Rui's research is focused on chemical tools to study the underlying mechanisms in infectious diseases, cancer, and neurodegenerative disorders. His current research interests cover the development of anti-infectious agents, prodrug chemistry and targeted drug delivery systems, design of covalent inhibitors, and design of chemical probes for activity and photoaffinity-based protein profiling. He has published over 150 papers, and he was the co-founder of the Medicinal Chemistry Division of the Portuguese Chemical Society. He is currently the president of the European Federation for Medicinal Chemistry and Chemical Biology. So Rui, I now will give the floor to you, and we are really looking forward to your talk today.
Thank you, Kathy, for your kind introduction. And it's a pleasure for me to be part of this webinar series of the EU-OPENSCREEN. So I will first start sharing the screen, the presentation.
Great.
I suppose that now is in the presentation mode.
Yes, that's perfect. Thanks, Rui.
And so let's start. So the topic today, as you said, is about chemical tools to target ferrous iron-dependent biology. This is let's say a currently hot topic regarding the importance of iron in regulating some of the biological phenomena in cells, and also the implication that is also relevant in terms of pharmacology. So my presentation will address, first of all, the basis of the ferrous iron-based pharmacology and the biology that is behind this topic, and then how to use this specific element to promote targeted drug delivery systems. So let's go to the first part, which basically is about targeting iron-2-rich environments, which endoperoxides. So, as most of you know, that iron exists in two forms in cells, iron-3, which is the most common one, and iron-2. The interchange between the two forms of iron is really important, because iron-3, in a way, is involved as cofactor of several enzymes, so it has an impact in the activity of enzymes, is also involved in other different biological phenomena. And iron-2, which exists in much smaller concentrations, is part of this balance, this redox balance, and when it is not controlled, it tends to overproduce reactive oxygen species, which then lead to cell death. So this is a very tight balance between the two forms. And in fact, when we go to understand how is the iron uptake and export controlled, so it is iron is usually transported across our body through the form of iron-3 together with transferrin, and once it binds the transferrin receptor, there is the endocytosis that leads to the transport of iron-3 to lysosomes. And lysosomes is let's say, the deposit of iron. Here, iron-3 is converted into iron-2 by step-3 protein, is exported to the cytosol by the divalent methyl transporter, and here iron-2 becomes part of what is commonly called the libel iron pool, where it controls several processes. It can go direct to mitochondria and be involved in, for example, in the inlet transport within the mitochondria, and then can be stored as iron-3 in ferritin. In the export process, the export is in the form of ferroportin transporter, which is controlled by epticidin, but basically iron-2 can also be reconverted to iron-3 and be part of what we call the autophagosome, and involved in the ferritinophagy in a way that controls the protein that stores iron in cells. So there is a very tight balance between iron-3, iron-2, and in many cancer cells, in fact, iron-2 is in higher concentrations. The exact concentration is still a matter of debate, but anyway, iron-2 can be considered as a unique signature for tumor initiation and development. But in fact, the understanding how the biology of iron-2 is important in terms to translate, for example, into pharmacology, starts with malaria. In malaria, which has a very parasite has a very complex life cycle, what is really important is that after infection of humans, the parasite goes to liver and then starts infecting the red blood cells, and the parasite can heavily infect red blood cells, leading to high concentrations of iron through hemoglobin, and the iron-2 accumulates in the red blood cells and becomes a target for therapy. Here, the concentrations that we are talking about are much higher than the ones that we talk about in cancer cells. But anyway, this was the beginning of how to use iron-2 as a pharmacological target. The way to target iron-2 was discovered in the 70s through a very interesting natural compound, artemisinin, which contains an endoperoxide, which chemically is called the 1,2,4-trioxane. And this endoperoxide can be triggered by iron-2, because the two oxygens are not chemically equivalent. So we have two different positions for the reaction iron 2, forming oxygen-based radicals, which then can undergo to rearrangement to carbon-based radicals, which then can be involved in the reaction with the peroxide targets. Interestingly, as would be natural with peroxides, EFMC can undergo what we call the phantom chemistry, which generates hydroxyl radicals, and in this way can be involved in also generating reactive oxygen species, also involved in cell death. So this compound is the source of very important reactions in cells, radical-based, in a way that that becomes a very interesting tool to study the biology where iron 2 is involved. So the first approach to use and develop tools to address the biology of iron 2 in malaria parasites was the development of multi-stage endoperoxide-based hybrid compounds. So the idea would be to couple an endoperoxide with another pharmacophore that could also be involved with a different stage of the parasite life cycle. And the basic principle was to use the endoperoxide to target the blood stage of the parasite, and to use a second pharmacophore, for example, to target the liver stage of infection by the malaria parasite, and use and connect these two parts through a linker that in a way could be used to modulate, for example, metabolism or aqueous solubility. And these are just two examples of this type of hybrid compounds that were developed with different linkers that have completely different physical chemical properties, also metabolic stability properties. And for example, we can modulate solubility by using amine that can be transformed into salts, and often in this way at different points to improve physical chemical properties. So interestingly, when we use these compounds in a model, an animal model of infection, of liver infection, and compare this with a standard compound that is used to study or to affect or kill the malaria parasites in the liver stage of infection, which is primaquine. And we were able to see that, first of all, our compounds were quite efficient in the simple cell model, but when translated into in vivo model, some of the compounds were quite efficient, even more efficient than primaquine, in blocking the liver stage infection. And when we use an animal model of infection for the blood stage, what we were able to see that these compounds were able to completely block the infection in the blood stage, one of the compounds, which translated in terms of the total survival of animals in the time scale of the experiments. So in this way, we were able to develop these tools that can interfere with the iron 2 biology, blocking the infection of the blood stage, but also appending another pharmacophore. We were able, in fact, to block two stages of the malaria life cycle parasite. Now, so once we had established this approach for the malaria parasites, which in fact they have a higher concentration of iron 2, the question would be if we could translate the same approach using endoperoxide to target the level iron pool in cancer cells. And the approach that we followed, in fact, was based in findings that had been recently reported. For example, when these close analog of artemisinin, which is artesinic acid, very well known as antimalarial as well, was found to be a potent compound against the colorectal cancer cells with an IC50 value of 2 micromolar, or this hybrid compound which contains this natural product, so thymoquinone, which in fact is involved in redox processes, also displayed a very interesting IC50 value against the colorectal cancer cells. So this triggered the development of a similar approach, now slightly different in a way that we appended a warhead that could react with proteins, with target proteins in cancer cells, and again using the linker for modulating metabolism and solubility. So basically the concept was to use the endoperoxide coupled with vinyl sulfone that could react with cathepsins in cancer cells, which are cysteine proteases that typically are overexpressed in several cancer cells, and also in some cases are also in higher concentrations in the tumor microenvironment. So basically the principle was again the same, using dipeptidyl vinyl sulfones, basically having the peptide sequence typically found for cathepsin inhibitors, using a linker to modulate physical chemical properties and the endoperoxide. And in fact, we were able to identify quite potent compounds against the colorectal cancer cells. And from here, we screened and generated a library of compounds basically by changing some of the amino acids in the sequence, removing a second amino acid, and of course, by changing the linker. Some of the compounds of this library are just depicted here. For example, we were able to see that having in the first amino acid either phenylalanine or homophenylalanine, we were able to generate compounds that were quite potent against several types of colorectal cancer cells, including those ones that contain mutated KRAS. And when we shortened the sequence by removing one amino acid, in this case, we were able to see that the compounds were quite toxic when tested against normal cells. And again, when we saw, we used the dipeptide hybrid compound that we found the highest potency coupled with high selectivity when tested against normal cells. So basically, we were able to identify compounds that were potent at the submicromolar range, high selective against the colorectal cancer cells. When we compare these results, for example, with the endoperoxide alone, clearly the endoperoxide was not able to generate the same level of activity. So basically, artemisinin was inactive in our assay. And for example, the dipeptidyl vinyl sulfone was not as active as our hybrid compounds, for example, when compared with our HIT. And we also tested and compared our results with selenomycin. And the reason for that is that selenomycin can, in fact, in a way, force iron to be sequestered in lysosomes, and in that way, it can deplete iron from cytosol and convert it into iron to generate in high levels of a reactive oxygen species. So in a way interferes also with the iron balance in cells, And in fact, it was interesting to see that selinomycin, while being highly potent against the colorectal cancer cells, was also highly toxic against the normal cells. So in terms of the therapeutic window, it's in fact highly toxic. We also studied these compounds against spheroid cancer cell models, so cancer cell stems, cancer cells. Basically, what we found is that the level of potency was maintained. So however, each compound continued to be highly potent and highly selective, so basically non-toxic, particularly when compared with selinomycin. So the next step was to translate these results obtained in cancer cells into an in vivo model. So what was used was the xenograft model for the colorectal cancer with a specific scheme of administration. Compound was given IP, and when we compared the tumor volume after almost 18 days of experiment, we were able to see that the volume of the tumor was substantially maintained when compared with the starting of the experiment, and particularly that difference was significantly high when compared with the vehicle that was given at the same time. So basically, we were able to show that using endoperoxide coupled with a system that can react with the cathepsins can lead to highly potent compounds against the colorectal cancer cells with in vivo efficacy. So basically, I think that to summarize this part of the talk, so clearly I think that the labile iron 2 can contribute to iron-dependent cell death, ferrocortosis, and infection in malaria. Importantly, the endoperoxide vinyl sulfone hybrids show to be active against mutated KRAS oncogenic colorectal cancer cells, and also displayed in vivo efficacy as a proof of concept. And of course, what with this generates is the possibility of using these endoperoxide-based hybrids to develop new therapeutic approaches for cancer. So this work now is being translated with optimized lead compounds, so that basically we hope to have a more advanced candidate in a shorter time. So the second part of the talk is about using iron-based or iron 2-based target drug delivery system. So the question here is that can we use this approach to develop what we call ferrous iron activable drug conjugates? And when addressing this approach, we go to endoperoxides that are using to treat malaria. So one of the classes of compounds that are under clinical trials at the moment are what we call trioxolanes, so a different type of endoperoxides. This compound here has been in clinical trials to treat mild malaria. And basically, again, the iron 2 coordinates one of the oxygen atoms of the peroxide function and activates the reaction. So basically what happens is iron 2 it can react with one of the oxygens, generates an oxygen-based radical, rearrangement to a carbon-based radical, which then can react with the parasites biomolecules. Of course, the two oxygens are not equivalent, so I only showed here one of the reactions, but basically the principle is identical. The only point to take attention here is that, in fact, oxygen 2 tends to be more reactive with iron 2 than oxygen 1. So basically, this is the main pathway. And what we have done in collaboration with Paul O'Neill from Liverpool was using this approach basically to have the possibility that after activation by iron 2 to generate a carbonyl-based compound that could act as cysteine protease inhibitor, and apply this concept to falciparum 2, which is the main or one of the main cysteine proteases that is involved in hemoglobin degradation by the malaria parasite infecting the red blood cells. So several compounds were, triaxolanes were prepared, and the basic idea was once they were activated by iron 2, they would generate it, for example, in this case, an aldehyde, or in this case, a methoxy methyl ketone, which are known to react with cysteine proteases. And in fact, so when we compared the IC50 value of the what we should call prodrug of the cysteine protease inhibitor, as you can see, the IC50 value against the enzyme is just moderate to poor. In fact, this is inactive, and once activated by iron 2, the compounds were shown to be quite potent inhibitors, particularly the aldehyde, but this is a trend that is commonly found as the inhibition potency against other cysteine proteases. Anyway, these results did not translate into efficient in vivo or in vitro assays to kill the parasites, and then we had to move in terms of the concept. So basically, what we did, we changed from triaxolanes to tetroxanes. Tetroxanes are another class of endoperoxides that has been used to develop antimalarial. So this compound has also been used, included in clinical trials at the moment. And the concept is, again, the same. Basically, we have two, in this case, two peroxide bridges, which are chemically equivalent. Activation by iron 2 generates first the oxygen, then the carbon-based radicals, which then can react with a target, but at the same time, it has also a ketone that can react with cysteine proteases. So the first approach that we used was, again, using tetroxane, pharmacophore with dipeptidyl phenylsulfone that would react with the cysteine protease falciparum 2, and the idea would to generate the inhibitor after the activation of iron 2 at the same time that the radicals could react with other targets in the protein. We did the activation in infected red blood cells. So it was found that the endoperoxide, the tetroxane, in fact, was quite rapidly activated to generate the cysteine protease inhibitor in less than 4 hours. So this was a quite efficient way of activating that was done both in a very, let's say, simple chemical system or in infected red blood cells. We also translated this concept from irreversible covalent inhibitors of falciparum into reversible covalent inhibitors of the cysteine of protease using nitrites. So basically the same concept again was to incorporate the cysteine protease inhibitor in or together with the tetroxane pharmacophore, and after activation of by iron to generate the cysteine protease inhibitor. In this case, in our libraries, what we found is that there was no difference between the prodrug itself and the cysteine protease inhibitor. Anyway, it was found that activation was quite efficient, slightly slower than in the previous case, but anyway, it was quite efficient. And what we found in terms of explaining why both prodrug and the cysteine protease inhibitor are quite efficient in inhibiting phosphopene-2 is related how both compounds can occupy particularly the S3 binding point pocket of phosphopene-2. When translated into the in vivo model for the blood stage infection, what we were able to see is that this compound was able to allow full survival of animals in the time scale of the experiment. And when we translated this into an analog containing a pharmacophore that was capable of blocking the liver stage of infection, basically the same result was observed. The animal survived throughout the time scale of the experiment. So we then decided to move into a different approach that basically was designed to allow a release of an actual payload through the iron to activation and subsequent beta elimination reaction. So basically the idea was to use the tetroxane as a trigger, a cleavable spacer that would allow the release of a payload after activation by iron 2. So basically the same process would happen, generation of radical depending on the oxygen that was reacting with iron 2, and then generating a radical in different parts, but anyway to generate the possibility of eliminating a cleavable linker and deliver the actual payload. For proof of concept, we use doxorubicin as a model drug. So this was developed by Diogo. What we did was to develop a simple process to generate the endoperoxide, the tetroxane. So oxidation of this diol, generating the hydroperoxide, generating the endoperoxide with different substituents in the adamantane. So basically, again, to modulate the physical chemical properties, and then through activation with the carbonate, we got the final prodrugs with reasonable yields. So activity in cells. So showing that in fact iron is part of the triggering process of these conjugates. We were able also to translate these compounds into one of these compounds into the in vivo study, and basically what we saw is that the conjugate showed a slightly better profile, in vivo profile, and so basically now we are currently optimizing these compounds to improve basically physical chemical properties as well their potency. So just to summarize, I think I hope that I have convinced you that the tetroxane-based drug conjugates are rapidly activated by ferrous iron. So they are excellent tools to study the biology and to develop pharmacological tools for cancer cells, in addition to malaria. And that this activation involves free radical species. So basically, that can also be involved in ferroptosis as mode of cell death. And of course, they have the potential to translate into in vivo potent and efficacious compounds. And just to finish, I hope that I have shown that tetroxanes and in fact other endoperoxides are unique tools to interrogate ferrous iron dependent biology that can translate into iron 2 dependent pharmacology. Hybrid compounds are excellent tools when we want to interfere in different points of the biology that is we are studying. And we used this system to in fact allow a selected delivery of toxic payloads in cancer cells, which translates into in vivo efficacy. I just, to finalize, want to thank to the teams that were involved in the work that was presented in the Faculty of Pharmacy at the Research Institute for Medicines, the medicinal chemistry tools, and particularly to the people that was involved in these studies, Diogo, Rita, Daniela, Rudy, and Joanna. Part of the phenotypic screening was performed at the Cecilia Rodrigues lab, and the in vivo studies by Manuela and the other people that was involved in the malaria studies. And thank you very much for your attention.
Many thanks, Rui, for this very interesting talk. I think we learned a lot now. I would suggest to go directly to the questions from our participants. I will start with the first one, which says, what is the source and nature of the free ferrous iron that appears to be mobilized during bacterial infections?
So the source that comes from bacteria infections can come from the host cells, and basically the similar mechanism to import the bacteria is similar mechanism to import iron to the intracellular environment, and in fact that can be used also to basically use the same approaches to use these tools to in fact kill bacteria.
Great, thanks, Rui. Another question, how do elevated ferrous iron concentrations promote cell growth and proliferation in cancer?
So one of the ways that iron 2 promotes the growth in cancer cells is basically to generate reactive oxygen species, which basically promotes cell death via ferroptosis. So that's one of the probably the main pathway to in a way that iron 2 generates cell death.
Great, that makes sense. Another question, until very recently, it has not been possible to study the ferrous iron specifically, largely due to the fact that it has much poorer ligation by small molecules or protein siderophores. Can you comment on this?
So basically, the question of iron 2 in fact as poor binding abilities in the what we call the labile iron pool in the cytosol, iron is as chaperones, iron 2 as chaperones, and in fact is as a weak ligation with glutathione. So there is an iron sulfur let's say weak binding that allows to keep the concentration of iron 2 in a, let's say, more controllable level. But it's true that iron 2 has a poor binding affinity. That is probably one of the reasons why up to now there is no, let's say, quite efficient way of determining iron 2 concentration, because the level of binding, we need very good chelators for iron 2 selective ones, because the difference of concentration between iron 2 and 3 is really high. And so, but in terms of what we want for activation, that is not a problem, because iron 2 is much more reactive than iron 3. And in fact, what we have done, and so this is a common procedure when developing endoperoxides, we need to compare the reactivity of these endoperoxides in iron 2, in the presence of iron 2 as well iron 3, to show clearly that is a difference of reactivity. And in fact, this activation just not will expand the answer, it makes it more broad. These endoperoxides, tetoxanes particularly, they are quite selective when compared with other divalent metals. So iron 2 is extremely selective in activation of endoperoxides.
Thanks, Rui. Another question about ferroptosis again. Is the labile iron pool necessarily elevated in cells that are sensitive to or actively undergoing ferroptosis?
Not necessarily. So ferroptosis basically is one of the main triggers is in fact generation of reactive oxygen species. They are typically over-generated in the presence of iron 2, but there is necessarily not a direct link. The reactive oxygen species can, in fact, be generated by the simple formation of hydroperoxides by oxidation of lipids, which they are not necessarily formed by iron 2. So there is not a direct link. And basically that's one of the challenges in studying the iron 2 biology in cells.
Great, thanks, Rui. I think we have time for a couple of questions more. So I will go directly to the next one. Which normal tissues and cells harbor elevated ferrous iron concentrations under conditions of normal iron homeostasis?
Could you repeat the question? Sorry.
Yes. Which normal tissues and cells harbor elevated ferrous iron concentrations under conditions of normal iron homeostasis?
Right. So typically, I assume that question is related to normal cells, not cancer cells. That is my understanding of
Yes.
of the question. So one of the organs which has a specific that is specifically involved in the iron homeostasis are the liver cells. So those are one of the type of cells that usually can have higher iron 2 concentrations. But anyway, the question is not about normal cells having higher concentration of iron 2. Is the difference between the levels of iron 2 and iron 3, which make this, when this difference is really high, that makes this possible to use this difference for, let's say, a pharmacological approach.
Great. Thanks, Rui. So, yeah, maybe a last question also would be, which iron regulatory proteins might be targeted to limit the unstable iron pool, and what effect would such intervention have in proliferative or neurodegenerative disease?
So the question of using this approach is to in neurodegenerative diseases is a tricky one, and the problem here is, of course, getting the compounds to go through the.
Rui, we cannot hear you anymore.
Can you hear me now?
Great. Yes.
Okay, so that's Zoom got self-life. So I was saying that the compounds, basically the challenge is to get the compounds through the blood-brain barrier, which might be one of the major challenges because these compounds tend to be highly lipophilic, and usually tend to have physical chemical properties that are not at the time being not amenable for this type of approach. In terms of if there are a difference of iron 2 and 3 levels in the brain, there are some reports that there are, but it's related. The problem is to get a very good tool to determine accurately the level of iron 2 in brains, and that is still a challenge.
Great. At this point, Rui, I would like to thank you very much for being here with us today. If our attendees have more questions, I'm sure they will be able to send us an email and I will forward to you. Thank you very much for being our speaker today and hope to see you soon.
It was a pleasure.
In our future webinars. Thank you very much.
Okay, thank you very much. See you all.
Thank you.
Bye.
Now, before we close our webinar, I would like to remind you that a survey will be sent out to you soon regarding our webinar today. So if you would like to know more about our training activities, you can also visit our website, www.EU-OPENSCREEN.eu. And yeah, have a nice day and see you in our next training webinar in February. Have a nice day.